Pues nuestra siguiente plática la va a dar el doctor Jim Leebens-Mack de la Universidad de Athens en Georgia, en Estados Unidos, y Jim nos va a platicar de sus estudios moleculares sobre la evolución de la poliploidía en los agaves, su cariotipo particular, que es muy interesante, que ya no mencioné, que es bimodal, 5 cromosomas grandes, 25 chicos, y la historia evolutiva de este grupo. Jim le va a dar la plática en inglés, pero no se asusten, si tienen cualquier duda, me preguntan y traduzco, y también las preguntas las vamos a traducir si las hacen en español. Thank you, Jim.
Thank you, Luis, for inviting me, and thank you all for being here. Pardon para my Spanish is not fit for speaking in public, I'm afraid. All right, so I'm going to talk about a couple of things that we haven't heard about yet today. First of all, the interesting cytogenetics of agaves, and actually yuccas as well, and everything that's in this agavoidy, or a good portion of the species that are agavoidy, have this interesting bimodal karyotype where we've got 5 large chromosomes and 25 small chromosomes. And so I'm going to delve into, well, how did this come about, and what's been happening since the origin of this karyotype. I'll pick up a little bit from where Luis presented on the phylogeny. I won't be talking a lot about the phylogeny of agave, but we have this large phylogeny that we put together based on mapping whole genome shotgun sequence data to a set of about 1,700 single copy nuclear genes, and gave us pretty nice set of relationships, very much in line with what had been previously published. But actually, right up around here, around Angustifolia, the Tequiliana genome, and some of the close relatives, not much, even with that much data, not very good support for relationships. That they're suggesting that this diversification is very recent, and maybe there's a little bit of gene flow going on between those species. So I'll talk a little bit about that. So first of all, work on this bimodal karyotype that I'll talk about extends from some work that was done by a former student of mine, Michael McCain, while he was a student in my lab. And he noticed that, well, really every member of this agavoidy, including hosta, which is native mostly to the Old World, it has this bimodal karyotype, and it's been largely preserved over the period of time since the origin of this group. These things out here do not have that karyotype. And so here we see that if we look at the genetic divergence between either duplicate genes within species, so when we're looking at hosta paralogs, the duplicate genes, we see, well, those are showing that Ks value that's here. And the hosta agave orthologs, the hosta chlorophytum orthologs, they are younger relative to that. So suggesting that, again, this arose here on the phylogeny. Here we've got the split between these bimodal karyotype things and everything else. Here we've got the origin of the bimodal karyotype, and here we've got the diversification of the bimodal species with this bimodal karyotype. Oop. So one thing that we were kind of interested in knowing, well, was this a simple, do we have a simple situation where one parent had these large chromosomes, one parent had these small chromosomes, they hybridized and gave rise to this bimodal karyotype? And no, this does not seem to be the case. So other folks have been looking at this, and really what seems to be happening is these large chromosomes are expanding as we've got lots of transposons that are just proliferating on the large chromosomes and not so much on the small chromosomes. We have no idea of what the mechanism for that is, but it's a very interesting observation, and I think it's worthwhile to go in and look at the mechanism of this. Something that would allow us that facilitate us of being able to do this is really good genome resources, a reference genome for agave. Luis mentioned that there's this genome that has been generated by the Chinese for a hybrid agave. Folks at Langbayo have been working on a genome assembly for Tequiliana. We, in parallel, group that led by Joint Genome Institute, in parallel worked on developing a very high molecular weight or highly resolved genome assembly, chromosomal genome assembly for agave. And so we put it all together, put the two teams together now, and we're working on a manuscript to describe these genomes and some of the interesting patterns of diversity in there. And I'll talk about a little bit of that today. So one of the things that pops out is that the last common ancestor, or the bimodal ancestor of agave and yucca and everything else in that bimodal karyotype, including hosta, a portion of the tree, it wasn't one parent, one diploid point, another diploid parent coming together to make a polyploid. But actually, the last common ancestor of all the bimodal karyotype things is a hexaploid. So here we look at any agave chromosome and compare it to any yucca chromosome, and we see, okay, the yucca and agave chromosomes line up very nicely. But then off of the diagonal, we see that there is large synthetic segments that are on other chromosomes. Broken up a bit, not quite as collinear as here, but large segments nonetheless that are on different chromosomes. And the other thing is that these chromosomes are kind of mixed between the large chromosomes. So this bit of the large chromosome has homologous segments in a couple of the smaller chromosomes that are in either species. If we look at the genome assembly that we did with JGI, we were able to split the maternal and paternal chromosomes, so it was a haplotype resolved assembly. And if we look at the two chromosomes, so here we're looking at one of the two haplotypes, here we're looking at the other haplotype, and we can see there's some really interesting structural differences between the two. So for instance, here in this haplotype that's shown on the bottom, we've got a big segment that was on chromosome 2 in this haplotype, and then in chromosome 3 in the other haplotype. And remember, these are two sets of chromosomes that are in the same cells in this agave Tequiliana plant. So that's very interesting, and again, something else that we're going to be delving into is the nature of the structural variation that we see. We go actually back to comparing the genomes with Allofolia and the two Tequiliana haplotypes. We see further structural variation, and again, mixing between bits of the large chromosomes and the small chromosomes. So for instance, here we've got a large bit that's chromosome 2 in haplotype 1, and it's over here on chromosome 6 and maybe a little bit on 5 in yucca. So very dynamic structural evolution in this bimodal karyotype. Now one thing is looking at the haplotype resolved genomes that we generated with JGI, we noticed, we compared that genome to the genome, a little bit more fragmented genome that had been produced by the Langbayo group, and we've noticed that one of the two haplotypes is generally much more similar to the Langbayo assembly than the other. And so here on the y-axis here, we're looking at genetic distance, and so up here, haplotype 2 is very different from the Langbayo assembly, whereas haplotype 1 is not so much. This other chromosome 2, on the other hand, we've got a mix, an exchange, and so part of the chromosome haplotype 1 is more distant from the Lang-Bio assembly, and chromosome 2 is more similar, and then that changes. And we went to check to see if this is an assembly artifact, and no, that this is other kind of structural variation that we see between the two haplotypes. So one thing that we're doing, we went and looked at, made gene trees and made kind of similarity trees for the genes that we were able to take from chromosome 1, chromosome 2, another chromosome 6, and we see that when we compare those to the sequences we got from some of these other species, generally the JGI haplotypes and the Lang-Bio haplotypes are forming a clade together, so they're Tikeliana. But which one is closer to the Lang-Bios I just showed? Haplotype 1 is closer for chromosome 1, haplotype 2 is closer for chromosome 2, and then we have some chromosomes where, one of the two haplotypes, haplotype 2 for instance, is more similar to micrantha than either of the Lang-Bio haplotypes or the other JGI haplotype, assembly haplotype. So Anastasia, who is while working as with Luis in Luis's lab, did some GBS analysis. She mapped the GBS data back to the JGI assembly, and then called SNPs. And actually, we put into this analysis the SNPs from the two haplotypes that we had from the JGI assembly. And first of all, we noticed that that Tikeliana generally has a very narrow gene pool, although everything is all of these individuals seem to be highly heterozygous, but they're all the same thing as Luis is talking about, except for this chromosome haplotype 1, it's closer to the Tikeliana cluster than some of these Augustifolia clusters, or espadine that mescal used for espadine mescal, the Augustifolia used for espadine mescal, but still it's pretty distinct relative to all the other genotypes that Anastasia had in her analysis. And then, as we might have expected by looking at that last slide, which one is further away flips for chromosome 2, and so we can looked at this for all of the chromosomes and we can see this sort of pattern. So the assembly that we currently have, and may be of use for your various studies, is in phytosome. Both of the haplotypes are in phytosome. You can go to these links and find them. But the one thing I would have you keep in mind is that these two haplotypes have are kind of mixed in terms of which chromosomes are closer to the rest of the Tikeliana and which one is further away. And we're working on a version 3 assembly now that's going to put all the chromosomes that have, all the haplotypes that have that are more similar to the other Tikeliana genotypes in one haplotype. Haplotype 1 we'll call it, version 3, and the other one will be the ones that are further away. All right, so I'm going to now switch gears. I just talked a little bit about this genome and about how we're using it, and the yucca genome as well, to better understand the ancestry of this this ancient hexaploid. But now, as I had in my title too, I was going to talk a little bit about gene losses and gene duplications on top of that ancient hexaploid event that we had in the last common ancestor of these species. And so, let me go back one here. One trait that I'm going to feature in talking about this is CAM, the ability to grow under very dry conditions, and so we think that CAM has arisen following this ancient hexaploidy event, has arisen 3 times independently. We're still kind of working on that, but that's our current hypothesis. And this is work done by a former student, Carolina Hayduk, who now has her own lab, is continuing this beautiful work at the University of Connecticut. So CAM, what CAM's plants can do is they take in their CO2 at night, fix it, as a fork carbon acid, put that fork carbon acid in the vacuole, and during the day bring that out of the malic acid out of the vacuole, and then have it feed the typical C3 carbon cycle. And we see that, okay, here's robust rubisco activity, that's what's doing photosynthesis, of course that's happening during the day, but Pep-C, this enzyme that is responsible for fixing this, the carbon at night, it's active during the day. As that works, we're building up malic acid, so the acidity is increasing through the night, and then in the morning, the malic enzyme and Pep-CK are liberating that carbon from the malic acid to feed the C3 cycle. So one thing we did in yucca was a very, and others have done this in agave, but focusing on yucca, we did a very broad transcriptomic analysis, collecting samples to do transcriptomics on every 4 hours through a 24-hour period. Pull out those genes. Pull out those genes that are showing time structure variation, and we notice that, and then compare them between a C3 species and a C4 and a CAM species. The CAM species has many more genes that are showing this time structure variation, and so let's go and talk about some of those. One is Pep-C, as we expect. It is expressed at higher levels in the evening as it may be priming to do its work, the protein do its work at night. And so one thing that's very interesting we notice is that there's two large families of Pep-C. One is the plant version, plant copy, and the other is the bacterial copy. Agaves generally are using this so-called plant copy. So these diverge from each other very early in the history of green plants. And it's one of these that has been used to do that nighttime fixation in agave. And actually, here's the ancient hexaploidy event. Which one is doing, duplicates of this gene were retained, and which one is doing the nighttime carbon, or this transcriptomic data just might be doing nighttime carbon fixation, differs in agave versus some of the other species, including agaves that we used to call Manfreda and Polyanthes. Whereas yucca allifolia, which is the CAM yucca, it is not using this gene copy, it's using this bacterial copy. So I'm just about done here. One thing, so again, we looked at lots of genes that were involved. These are a set of kind of canonical CAM genes. And when we compared their gene expression patterns in a C3 species, yucca species, Yucca filamentosa, and a CAM species, Yucca allifolia, and we asked, okay, what's happening in these gene families? Do we see a change in expression? Do we see increased expression? Or do we see a change from no time structure, the transcript being produced the same amount throughout the 24-hour period, versus a shift to time structure? And both of these things are contributing to what CAM in allifolia, in addition to gene duplications that contributing to the CAM. So my takeaways are then, all agaves, yuccas, and related genera are ancient hexaploids, so keep that in mind as you're analyzing the data from these species. Persistence of there's a persistence of this bimodal karyotype in face of, over time, in face of change in the structure of the chromosomes. Gene duplications and polyploidization have contributed to functional innovations, including the evolution of CAM. And then keep an eye out for the publication on this joint Mexican-U.S. investigation of Tequilianas, revealing this complex pattern of genome evolution. So I think I have time for one or two questions. I'd be happy to take them.
Y tenemos tiempo para un par de preguntas y si quieren las puedo traducir. ¿Algún valiente? Nadie va a preguntar.
Es que en la penúltima diapositiva, el esquema de abajo de la derecha, estamos hablando del metabolismo. Es que no alcancé a leer.
Podemos ver la
Which one?
una más atrás.
No, abajo a la derecha está el esquema. Está bien,
Pero ¿de qué quieres que le diga? Okay, she wants to ask you about the CAM metabolism in this slide.
Yeah. So these are some of the genes that are responsible for metabolizing carbon. Carbon comes into the cells at night. The CAM species are opening up their stomata at night, bringing in carbon dioxide then, and it's then being fixed as a 4-carbon acid, malic acid, stored in the vacuole, and then during the day, that 4-carbon acid comes out of the vacuole and 1 carbon stripped off to feed the C3 cycle without the stomata open. So C3 species, they close their stomata at night. They have them open during the day to bring in CO2. But as in the process, they're losing water. So this is a mechanism to increase water use efficiency. So that's what CAM is, what's going on with CAM. And the point I'm making here is, okay, we have a set of genes that both the CAM and the C3 species have. What's going on to make the function of some of these genes shift from just doing basic work all throughout the 24-hour period to being upregulated at the time that they're they're doing work as part of this CAM system. And again, we've got both increased expression sometimes, so we're going from rather low expression levels to higher expression levels for some of these things. So that expression levels might be changing for some of these genes. And at the same time, we might be shifting from a one-time structure. So in this case, this is time structured, but the pattern of variation through the 24-hour period is different for the allofolia species. In other instances, we go from a flat, no time structure at all, to clear time structure in the CAM species.
Thank you, Jim. We have to move on, but maybe later they can ask you.